Welcome back to the deep dive. Today, we're looking at something that honestly it fundamentally shifts how we think about fighting cancer. We're talking about a personalized immune system hit squad.
Yeah, we're diving deep into the world of CAR-R and NK cell therapy.
And this is such a critical deep dive because the sources we've looked at, they show this field is just moving at warp speed.
It really is. It's accelerating out of basic research and, into clinical reality. We're looking at data that positions CAR-NK as a potentially safer, really targeted, and just immensely powerful alternative.
An alternative to traditional chemo and even the sort of established CAR T therapies.
Exactly.
And the scope is already just massive. I mean, we're talking about a therapeutic area with over 150 trials running globally right now.
Which is incredible.
So our mission today, I think, is threefold. First, we need to understand the clever science behind giving human immune cells this specialized super site. Then we have to grasp why the manufacturing is such a huge bottleneck, both logistically and financially.
It's a big one.
And finally, we need to weigh the risks against these groundbreaking success rates we're seeing in the latest trials.
And if you take just one thing away from this whole deep dive, let it be this key distinction. CAR-NK is about customizing natural killer cells, not T cells.
Right. Not T cells.
And that fundamental difference means the attack comes with a far lower risk of the patient's body having a severe rejection freakout, especially when you're using donor cells.
Okay, let's unpack that. The core mechanism. We should probably start with the soldiers themselves. What is a natural killer, or NK, cell? And what exactly does the CRR kininogen receptor do for them?
So natural killer cells are fascinating. They're like the shock troops of your innate immune system. They just patrol your body looking for anything that seems stressed or infected.
Or cancerous.
Or cancerous, exactly. And the crucial thing is, they don't need to be trained. They act immediately. The problem is sometimes cancer cells get really good at hiding their distress signals. The NK cells just miss them.
And that's where the CRR comes in. It's like it overrides the cancer cell's camouflage.
Precisely. The scientists genetically engineer these cells by the sources say stitching on a GPS.
Yeah.
And that GPS is the CR.
It's an artificial receptor, right? Chimeric.
Exactly. It's built from multiple biological parts that don't normally go together. It allows us to direct these naturally powerful cells with a surgical precision.
So the CIR is basically a homing signal.
Correct. It's designed to lock on to specific tags antigens that you see all over certain cancer cells, like CD19 in blood cancers or HER2 in some solid tumors.
And once it locks on...
Once it locks on, it triggers the NK cell to do what it already knows how to do, to deliver that powerful death punch.
What's really remarkable, and the sources keep emphasizing this, is the speed. The comparison they use is a hit squad, not chemo sledge.
It's a powerful image, isn't it?
Yeah.
It speaks directly to the advantage over chemotherapy, which is just systemic poison. With CAR-NK, the sources confirm you can see the effect in days, maybe a week. The tumors can just vanish. And because they're using donor cells, you can scale production, and the recipient's body is way less likely to reject them compared to donor T cells.
Okay, now we have to get into the how, because you can't really appreciate the treatment without understanding the immense technical process required to build this CAR-NK army. This part explains the cost and the difficulty in scaling it up.
It really is a multi-step factory process, and it has to operate under just extreme sterility and precision. The sources break it down into three main stages, starting right at the drawing board.
Okay, so give us the breakdown. What's the first major challenge?
Stage one is designing and building the blueprint. This is creating the CAR-NK gene itself. You need specific parts, something to grab a tumor, an anchor, and an internal signal.
A firing pin.
That's a great way to put it. It's a firing pin that tells the NK cell, I found the target, now activate. If that blueprint is even slightly off, the whole thing fails. The cell won't fire.
So once you have this perfect genetic blueprint, how do you get that massive piece of information inside a tiny little NK cell?
And that's stage two, vector manufacturing. You can't just inject the gene. You need a delivery vehicle, a vector. This is usually a modified virus.
Like a lentivirus.
A lentivirus for a permanent, stable integration, or maybe an AAV, which is a bit safer because it doesn't permanently wedge itself into the host DNA.
So we're essentially reprogramming viruses to be microscopic delivery trucks.
That's exactly right. And making those trucks is incredibly complex. You have to grow specialized cells in a clean room, feed them the core gene, plus all these other helper genes the virus needs to assemble itself.
I'm just imagining the quality control on that. It sounds like a nightmare.
It is. If any of those helper elements are wrong or if there's any contamination, the whole batch of delivery viruses, it just fails.
Wow. Okay, so you've made the delivery trucks. What's stage 3?
Stage 3 is acquiring and transducing the NK soldiers. You need high-quality NK cells. You can get them from donor blood or, and this is the future, from these highly scalable induced pluripotent stem cell lines, iPSC lines.
The off-the-shelf supply.
The off-the-shelf supply, exactly. And you have to keep them happy, active. You culture them with activating signals like IL-2 and IL-15 to make them receptive. Then you mix them with the delivery viruses.
And how do the scientists actually know if this whole incredibly intricate process worked? This feels like the point of no return for the whole batch.
This is the critical quality check. If the scientists were smart about their design, they spliced in a green fluorescent protein, a GFP tag, right alongside the CAR gene.
Ah, so if the CAR gene gets in, the cell literally glows green.
Precisely. They wait about 48 hours, look under a special microscope, and the successful cells, well, they should glow.
And what's a good success rate?
They're looking for 50 to 70%. If it's lower, say 5 or 10%, the batch is useless. The whole effort fails. That is a major manufacturing risk the sources mention over and over.
50 to 70% glow success, a very clear metric. And once they have those positive glowing cells, then it's just about mass production.
Yes, the final step is expansion. They sort the positive cells and feed them growth factors until they have billions of them. Then they're frozen, shipped, and thawed right at the patient's bedside for infusion. That's the journey from a computer design to a living drug.
That complexity is just staggering. But let's move to the critical question. What are the risks? This is powerful medicine, but it's not harmless.
You're right. And despite the safety advantages over CAR-T, there are specific dangers. The biggest one is called off-target cytotoxicity.
The GPS misfires and hits healthy tissue.
Exactly. The binder might accidentally hit a normal cell. For instance, if you're targeting HER2 for breast cancer, well, HER2 is also found on heart cells.
So you could cause heart damage.
You could cause severe myocarditis. But here's the crucial nuance from the sources. Because NK cells naturally die off much faster than engineered T cells, that damage tends to be short-lived.
That's a really interesting point. If the damage is short-lived because the cells die quickly, doesn't that make the treatment itself less durable? How do they deal with that?
That is an excellent question and a major challenge. Durability is a concern for sure. Labs are tackling this by giving repeated high-dose infusions, maybe weekly, and also by engineering the NK cells to produce their own activating signals.
So they can sort of self-boost their own lifespan.
Right, to keep them potent after infusion.
Okay, that makes sense. Now, what about that immune system overload? We always hear about cytokine release syndrome, CRS, with CAR-T therapy. How does CAR-NK compare?
CRS is definitely a risk. It happens when the cells kill the tumor so fast that this massive inflammatory signal gets released. You get fever, low blood pressure.
But is it as bad as with CAR-T?
The good news is no. The CRS in CAR-NK trials is consistently reported as significantly milder. The sources specifically say less IL-6 storm, which is that dangerous cascade that often puts CAR-T patients in the ICU.
So less inflammatory risk. But what about the body just rejecting the donor cells?
That's the big efficacy challenge. The recipient's immune system still sees them as foreign and tries to clear them out. It's not the severe graft versus host disease you see with T cells, but they can get eliminated quickly.
Which means you need bigger doses or more frequent ones.
Exactly. Large, high-frequency doses or careful HLA matching to make sure the treatment has enough time to actually work.
And we touched on manufacturing risk, but what about the worst-case scenario from the vector itself? Can the delivery truck cause a new problem?
You mean insertional mutagenesis, basically causing a new cancer by disrupting a vital gene.
Yeah.
That's a risk, mostly with integrating viruses like lentivirus. But the industry trend is moving toward non-integrating viruses, like AAV, specifically to dodge that catastrophic long-term risk.
Okay, so we've weighed the risks. Let's shift to the rewards, the efficacy data. Are these cells actually winning the fight?
They're showing immense promise, especially against blood cancers like leukemia and lymphoma. Phase 1 trials are showing complete remission rates of 20 to 40%.
And this is in patients who have failed other treatments?
That's the crucial detail. These numbers include patients who had already failed conventional chemo and CAR-T therapy. For a lot of them, CAR-NK is truly the last best hope.
What about solid tumors? Breast, lung, pancreatic cancer, the notoriously difficult ones?
That's still the frontier. The response rates are lower, maybe 10 to 15% so far, But the sources show rapid improvement with combination strategies.
So not just using the CAR NK cells alone.
Right. They're layering them with checkpoint inhibitors,
Mhm.
like anti-PD-1 drugs. It's like taking the brakes off the existing immune system while the CRNK cells launch their attack. That synergistic approach is really boosting those response rates.
And to circle back on the safety profile, the sources seem almost unanimous that this is still safer than chemo. And crucially, no bad neuro stuff yet.
And that is a massive clinical advantage. The severe neurological side effects you sometimes see in T-cell therapy just seem to be absent so far in the CAR-NK trials. To have a therapy this powerful that avoids all that, it's a huge leap forward.
So with all these trials running, what kind of off-the-shelf treatments are starting to emerge?
We are moving towards standardization. You have drugs like
Sci-fi
NK.
development in the source material. The idea of real-time tracking.
Oh, this is truly game-changing, not just for therapy, but for diagnostics. Some of these teams are engineering the NK cells to produce light using a tag called nanoluke.
Nanoluke, that's a luciferase, right? Like a firefly. So we're not just making killer cells, we're making glowing killer cells.
That is exactly the aha moment. They mix the nanoluke gene right into the CAR tail, and it acts as this non-invasive internal beacon. It allows for real-time tracking of the cells inside the patient's body.
That's just astonishing. So instead of waiting weeks for a CT scan, doctors could see if the drug is working almost immediately.
Immediately, you can see where the cells are going, if they're reaching the tumor, how many are surviving. You can paint the picture. Imagine a scanner passes over the patient, and their chest lights up as the tumor is saturated with these glowing killer cells.
And then as the tumors vanish, the light starts to fade.
Precisely.
And how is this actually tracked?
It's done with specialized imaging rigs, or even just highly sensitive benchtop scanners that can detect that faint light through the tissue. It gives doctors immediate, actionable data on drug efficacy without invasive biopsies or delayed scans.
This entire deep dive, it paints a picture of CAR-NK as this highly sophisticated, specific, and maybe the safest immunological leap forward we've seen yet. It's a genuine option for patients who've run out of road.
It really is. It capitalizes on the raw power of the NK cell, but mitigates the risks we see with its T-cell counterparts. The manufacturing is still the barrier, but the clinical rewards, especially that safety profile, make this one of the most exciting fields to watch right now.
So to leave you with a final thought to explore, the sources suggest that this real-time nanoluke tracking isn't FDA approved yet, but they're projecting maybe a 6-month window before it enters common practice, just because the pace is so fast. So, if, in the next year, we shift from invasive biopsies and MRIs to these personalized real-time diagnostic light shows, what does that do for the patient experience? What does a glowing readout mean for the emotional toll of cancer treatment compared to the uncertainty we have now?